everyone, and welcome to the Science of Childhood Cancer lecture series, hosted by the St. Jude Comprehensive Cancer Center. I am Paul Northcott, an associate member here at St. Jude in the Department of Developmental Neurobiology and affiliated with the Neurobiology and Brain Tumor Program. It is my great pleasure today to welcome our speaker, Dr. Brad Bernstein from the Dana-Farber Cancer Institute in Boston. I actually first met Brad at a Keystone meeting during my PhD. I'm sure he doesn't remember. When he came to my poster and grilled me on the role of chromatin modifier alterations in medulloblastoma. In recent years, my lab has been working with Brad's on single-cell applications in childhood brain tumors, and it is truly my honor to introduce his lecture here today. Before we begin, I'd like to note that you can submit questions at any time during the talk using the chat function and send questions to all panelists. If there are additional questions, Dr. Bernstein has kindly offered to stay on for some time at the top of the hour to further discuss his research interests. Now I'm delighted to introduce our speaker for today. Dr. Bernstein is the chair of cancer biology at the Dana-Farber Cancer Institute, where he holds the Richard and Nancy Lubin Family Chair. He is also the director of the Gene Regulation Observatory at the Broad Institute, a professor of cell biology and pathology at Harvard Medical School, and an investigator at Harvard's Ludwig Institute. Bernstein received his BS from Yale University and his MD PhD from the University of Washington before completing a residency in clinical pathology at Brigham and Women's Hospital and postdoctoral research at Harvard University. He launched his independent research program in the Department of Pathology at Mass General and Harvard Medical School in 2005. He was promoted to full professor in 2013 and awarded the Bernard and Mildred Caden Endowed MGH Research Institute Chair in 2016. During the past year, he has transitioned into his current role at the Dana-Farber. Bernstein's research focuses on epigenetic gene regulation. His lab studies how gene activity is controlled by non-coding regulatory elements such as enhancers and by the way the genes are packaged into chromatin. Bernstein's work is notable for the discovery of bivalent domains, a signature chromatin state consisting of opposing histone modifications that poise master genes for alternate fates. His characterization of bivalent chromatin and associated regulatory factors in stem cells was a key early demonstration of the mechanistic impact of chromatin on mammalian development. Bernstein's subsequent work as a leader of the NIH's ENCODE consortium revealed that the vast non-coding portions of the human genome, which had been previously dismissed as junk, are in fact packed with sequence elements that control gene activity. His second major area of contribution is cancer epigenetics, as we'll hear more about today. He showed that DNA methylation can activate oncogenes by disrupting genomic insulators, an entirely unexpected discovery given that methylation had been so closely tied with gene repression. This finding explains how certain tumors can sustain potent oncogenic signaling in the absence of canonical mutations. His group has also uncovered epigenetic mechanisms that underlie tumor cell self-renewal, drug tolerance, and immune evasion. Bernstein's honors include many, such as an Early Career Scientist Award from the Howard Hughes Medical Institute, an Elizabeth and Michael Ruan Endowed Scholar in Endocrine Pathology, a Career Award in the Biomedical Sciences from the Burroughs Wellcome Fund, the NIH Director's Pioneer Award, and many more. Without any further delay, I'd like to turn things over to Brad and look forward to his lecture on epigenetic mechanisms of tumorigenesis.
Great. Thank you, Paul. Very kind introduction, and I guess of some memories. It's since that Keystone meeting, it's been a lot of fun watching your career absolutely blast off over the last years. Anyway, good to be back. I was telling Paul that the last work trip I took was actually to Memphis to do the Danny Thomas Award. It was a great visit, and I'm sorry that I'm doing this one in person because I would love to be there today. So I have, Paul talked about my titles. I have some disclosures here, and some of the work I'll talk about is actually funded by Calico. I want to try something a little different today than usual. It's a new talk. I want to synthesize our latest thinking in about the epigenetic clock and its role in cancer. So I'm going to be intermixing published work from us, published work from some other scientists in the field, and perhaps a bit of speculation. So I hope you enjoy it. It's the first time I've given the talk, so there may be some holes, and feel free to follow up with questions. So, the big picture that I want to sort of argue to you today is that there is this sort of monotonous, unstoppable process whereby DNA methylation is gradually lost over gene deserts as cells accumulate divisions. Every time a cell's divide, a little bit of methylation is lost. And make the case that this hypomethylation is really evident in both aging tissues and in cancer cells, both of which have divided a lot. And this is, you might think of this as an epigenetic clock or a mitotic clock. I'll touch on this, how it might relate to an aging clock. Functionally, we think that hypomethylation is actually tumor suppressive. It's a little counterintuitive since tumors have a lot of hypomethylation, but I'm going to try to make the argument that the hypomethylation is a tumor suppressive function. It's turning on transposable elements, triggering immunity, and changing the 3D genome topology in ways that suppress tumor progression. Then finally, I'm going to talk about how tumors might adopt strategies to overcome this barrier, and I'll give you one specific example from our recent work. So DNA methylation is really a bona fide epigenetic mark in that it's really transmitted through to the daughter strands as cell divide. It's kind of like you copy your sequence, but when there's a methyl group, in principle, DNMT1 comes along and it'll find places where the methyl groups are, and it'll put the methyl group on the newly synthesized strand as well. So this is sort of that idea of epigenetic inheritance through mitosis. Does a great job of doing this in regions where there are like dense methylated CPGs. Lots of CPGs are methylated. They are efficiently maintained during replication. It does a less good job of doing this in regions where the CPGs are isolated. If you have a methylated CPG in sort of a desert with low CPG content and there's no other methylated CPGs around it, for various reasons, that's we know mechanistically it's often lost. It could be there's less methylation signal to maintain it. It could be these are late replicating regions, so the machinery is not as primed to copy. It might be that chromatin state is important for copying methylated regions, and the chromatin state is not as well declared in those regions. But point is, there's mechanistic basis for why, as cells divide, those isolated CPGs that are methylated will gradually lose their methylation. Now, initially, actually, most CPGs in your genome are methylated, with the exception of CPG islands. So those isolated CPGs start out methylated. This is a picture I borrowed from Ben Berman in a very nice paper from 2018. He shows sort of across 30, 40 megabases represented region. The yellow is methylation, and you can see this whole region. CPGs are pretty well methylated, excepting CPG islands. But generally, if you look globally at this kind of resolution, you see methylation in newborn T cells. But when you look at a centenarian's T cells, you can see these blue here and these big regions of blue, and those are regions that are losing their methylation. You see the same thing in a T cell leukemia. These regions have lost their methylation, and in fact, these regions that are bluish, these are low CPG density gene desert regions, and these big areas of hypomethylation are sometimes called hypomethylated blocks. We're very familiar with them from cancer, but I think Ben did a nice job of showing that this is happening in aging cells as well, not just cancer cells. And you can see this newborns. Actually, it happens during fetal development even, so gradually losing it, but then it really drops off between sort of a newborn and like somebody who's elderly, or between fetal and adult. You see this in almost all tissues, this gradual loss of DNA methylation in these deserts. Okay. So I want to make the argument that this is like a mitotic clock. Oh, actually, one more data before I get to there. I want to show you that the tumors, most tumors have global hypomethylation throughout these same regions. This is basically showing you a measure of hypomethylation in all these epithelial tumors and other kinds of tumors. Very few tumors don't have hypomethylation. All these tumors have really lost methylation in those same desert regions. They're all hypomethylated, so really hallmark of cancer. Okay, but as I said, I want to make this argument that this hypomethylation is a function of cell division, that the CpG methylation is lost as cells divide. Sarah Johnston in the lab, when she was in the lab, did a simple experiment. She passaged fibroblasts. So cells dividing, and sometimes you think of CpG islands might get methylated when you passage cells in culture, but we're not looking at those CpG islands. We're looking at those gene deserts, the CpG sparse gene deserts. And what you can see in this picture here is that those same sort of regions, there's hypomethylated blocks, and by passage 40, they're more hypomethylated. And this is just another way of showing that same thing here on the right. So, this really is a mitotic clock. As cells divide, there's a mechanistic basis. You don't do a great job of keeping the methyl groups there. They're gradually lost and lost and lost. And if you find cells that are aged or tumor that have accumulated many cell divisions, they have hypomethylation. Now, here's a little bit of a trick. Probably a lot of people have heard about the epigenetic aging clock from Horvath, Hannum, and others. It's a little bit of a different thing. The aging clock, these colleagues have looked across different ages for individuals, and they've sampled CpGs across the genome, and they have a couple hundred CpGs that if you integrate them in some way and get a metric based on those CpGs, you can kind of predict someone's age. It's different for different tissues. For blood, you have a clock, you have a different clock for brain. But it's pretty amazing that the correspondence between the score on that DNA methylation epigenetic clock metric and the age of an individual, it's just really spot on. You can predict or tell somebody's age basically within a few years based on methylation of their tissues. But the epigenetic clock, the aging clock here, people haven't really thought about that this is hypomethylation because the methyl groups are all over the place. They're not all in hypomethylated regions. But I want to speculate that probably this aging clock sampled in tissues is actually some composite of this mitotic clock. How many times the cells are divided in those tissues? It's complicated because there's stem cells and differentiated cells and different cell types. It's complicated because there's probably greater inflammation in elderly individuals that feed back and change methylation further. So the aging clock probably has multiple components. But there is a component, I think that mitotic clock component is a very special one because it is counting cell divisions. And I think for that reason, it's a critical and maybe deterministic component of the aging clock. And as I'm going to get to, I think it's a functional component with an important functional significance, the mitotic piece of the aging clock, okay? So to make that case, I'm going to jump to 3D genome organization for a little bit because methylation profoundly changes how the DNA is organized in the nucleus and its regulation. So if you'll bear with me, there's been a lot of work lately studying how that there's 6 feet of DNA, right? It's in every one of your cells, and it's got to wrap up and fit in the nucleus, and it's got to be properly folded up such that just the right regions can be accessed, and it can be regulated in the right ways in different cell types. You have multiple layers. It's organized hierarchically. You've got methylation, as I told you. You've got the wrapping around the histones, nucleosomes of about 150 bases. You've got loops formed between enhancers and promoters. You've then got sort of these higher layers or long-range structures. There's these topologically associated domains or TADs, which are sort of regions of hundreds of kilobases to maybe a megabase of genome that are self-interacting. They have boundaries flanking them. I'll show you more with that in a second. And then there are these compartments where sort of open active TADs come together with other active TADs and closed compact TADs come together with closed TADs. How do we know all this? This is nice pioneering work from Decker and Aiden and Wren and others. Proximity ligation methods, and I'll touch on those quickly, are sort of the mainstay, the anchor of this work. Basically, in proximity ligation, you take the nuclei and you fix them. So the chromatin is fixed and the conformations are fixed with formaldehyde, and they stay as they are. You then restrict with an enzyme to cut the DNA. and then you religate the DNA in situ, still in that native conformation. And there'll be a lot of religations to the same place you cut, but occasionally you got religations to other DNA that happen to be nearby inside the nucleus. And then what you do is you take that DNA and you sequence everything. And this is kind of the Hi-C method. And what you get is sort of a map. You measure contact frequencies. How often do you get ligation events occurring? And that tells you about something being in contact in a cell. This is what the data look like. Here's a chromosome map of a Hi-C map for chromosome 7. It's a pairwise map, so it's really showing chromosome 7 on the horizontal axis and chromosome 7 on the vertical axis. You see a nice red diagonal stripe. That's because regions that are close on the linear DNA, as you might imagine, are close in 3 dimensions, right? Because they can't be that far apart. But there's some other structures you can appreciate from Hi-C maps too. If you zoom in on that diagonal, you realize that there's a series of sort of triangles or steps. And what those steps are is the TADs. These steps are, on the order of 100 kb to megabase, and each step contains, corresponds to DNA that cross-link that will religate a lot to itself within a region. But the boundaries, the edges of these triangles here, are boundaries that prevent religation events across them and are basically telling you that you're separating, partitioning or TADs. And so these are these boundaries between these topologically associated domains. I like to think about TADs as sort of the functional unit of our genome, right? You've got these pieces of self-interacting regions where enhancers can act on genes and things like that, but they can't cross TADs, and they have a hard time contacting the neighboring TAD. There's another layer too, and this is these compartments. The compartments are appreciated in this. So you see there's a plaid, looks like a plaid shirt, the Hi-C maps. And the reason is that there are long-range contacts made between like chromatin. There are TADs like this one here on the top that are openish. And you can see that this open TAD makes some weak long-range interactions with other open TADs, right? So open compartment, big open regions sort of gently come together. And I'll give you a visual picture so you can appreciate why that happens a little better. Conversely, closed regions here, you can see kind of interact more with other closed regions. And if you put an eigenvector, calculate an eigenvector across these long-range interactions, you basically get 2 compartments. There's probably more, but at a gross, and I'll touch on that a little bit, but at a gross scale, there's 2 compartments. There's a compartment of open chromatin shown here in blue, compartment A, euchromatin, and a compartment of closed heterochromatin. The compartment A is more the GC-rich euchromatin, and the compartment B is that GC-poor heterochromatin. So Sarah Johnson in the lab wanted to, working with Martin Ari and Alejandro Reyes, wanted to understand how these topological structures, how might they be altered in human tumors? So we took a cohort of sort of normal colon and of colon tumors, and we performed Hi-C on, they did a lot of Hi-C on a lot of samples. And I'll show you really the most, let's see. The TADs didn't change. Those TADs, remember I told you this, the discrete units and the boundaries, they changed very little. It's rare. There are some important changes, and I think I talked about them when I was at the Danny Thomas. I don't have time to go into that. There are some changes, but they're sort of rare and in between. But what's really striking is the changes in the compartment structures. And this is just a plot here on the right I'm showing here, where we've calculated a differential in compartment structure between colon tumors and normal colon. And the red heat are regions where you get gaining interactions. And basically you can see that these compartment B regions are starting to interact with compartment A regions more in the tumor. So there's a global breakdown of this very nice partitioning that we normally see between compartment A and compartment B in the tumors. And that's what I'm going to spend a little time trying to explain what we think is going on. It's probably easier to think about all this as a model of how the chromosomes are folding up. And fortunately, we have his colleague Bin Zhang, who's a computational biologist at MIT. And Bin has a very nice way of developing polymer models of the whole genome, how it's folded up at low resolution, but nonetheless, the genome, how it's folded up. He simulates these models in silico and builds out models. Now, what he can do is he can use that model and he can calculate what the Hi-C map would look like for that simulation. And then you can ask, does that Hi-C map he just calculated for in silico model, does it look like the one we just calculated for normal colon? And then you can keep iterating until he gets something in silico that looks like it could explain what we're seeing in the Hi-C maps. And when he does that for normal colon, he gets this picture, a polymer picture of sort of low resolution chromatin strands. And hopefully you can appreciate that there's a nice partitioning between compartment B in yellow, that's on the outside. Remember, that's the heterochromatin, and compartment A on the inside, which is the euchromatin, the active stuff. This makes a lot of sense because we know that the silent stuff, the inactive stuff, moves to the periphery and is associated with the lamin. Here actually is an electron micrograph of a nucleus of a normal colon epithelial cell, and you can appreciate some of those features. You can appreciate sort of the nuclear membrane on the very outside of the nucleus here, and then you can see that juxtaposed against the nuclear membrane is dark staining, electron dense heterochromatin. This is very dense stuff. But all throughout the nucleus is euchromatin, right? The interior of the nucleus, it's the fluffy, loosely packed euchromatin, right? This is the compartment A stuff on the inside. So really, this polymer model matches up pretty nicely with the electron microscopy and with prior knowledge and what we would have expected. Here, just showing the same thing again. We can see this on multiple nuclei. Here's the model for normal colon. We can do exactly the same thing, though, for the tumors. When we do the simulation on the tumors to find polymer models that match the tumor Hi-C map, we see something very different. Now we see that a lot of this, the B chromatin, the yellow stuff, is no longer peripheral. It's moved interior, and there's sort of a disorganization that you see in tumors. And the electron micrographs for the tumor is actually quite complementary and consistent with that, right? You still see dark staining around the periphery, heterochromatin, but now you see a lot more of the dark staining heterochromatin moving interior. So there's this real shift in the compartments where this real dark, compact heterochromatin has moved to the inside of the cell a little bit. So a profound architectural change. And we think it relates to the hypomethylation. So I'm going to go back to the hypomethylation. I'll tell you two pretty interesting things. One is, remember I told you there's those big hypomethylated blocks? And here's just a picture of sort of the methylation levels in normal colon green and in colon tumors purple. And you can see that there's hypomethylation in the tumors. And it's really quite stark how it really just sits across this region, right? This is the block of hypomethylation. Andy Feinberg and others have described this. The block corresponds perfectly to a TAD. Here, this little triangle here is the topological domain. And it's the whole, when you start hypomethylating, you hypomethylate the whole topological domain. And if you look across the genome, you see that these hypomethylated blocks, they correspond to the current B compartments. And there's some shifting in I compartments, but really for this purpose, I think you can think of I as being very similar to B. It's a heterochromatic compartment, and it's got profound hypomethylation in the tumors. You see this genome-wide, basically perfect correspondence between hypomethylation and the B and maybe a little bit of I compartments. Not only is there correlation, though, we think there's some causation here, and I think there's a directionality that I'll show you. First, I'll tell you that if you take a TAD or look across TADs and you ask how hypomethylated are they getting in the tumors, the TADs that get the most hypomethylated, this is the bottom histogram here. The bottom histogram, they're more B-like. They're shifting left. They're becoming more compact. Okay, so as you hypomethylate, they're more compact. Okay, still it's correlative, but this might be a little causation here in the middle. If you take colon cancer cell line and you treat it with demethylating agent and you sort of force a loss of methylation, force of hypomethylation. You see new TADs becoming hypomethylated. And when those TADs become hypomethylated, again, shown in the bottom, the ones that lose methylation, those TADs specifically become more B-like. So they're shifting to become more compact, okay, in association with the hypomethylation. And as I showed you before, we think they're moving away from the periphery, they're moving anteriorly, and they're compacting as they get hypomethylated. And I can tell you this shift is associated with repression. So you might say, oh, it's moved away from the peripheral heterochromatin. Maybe it's getting de-repressed, but actually it's getting even more repressed. As you lose this methylation and become hypomethylated, you become organized within really repressive histones. Histone H3K9ME3 and histone H3K27 methylation, we think, are doing a pretty good job of keeping these things silenced as they move interior. So here's a model of what we think is happening. This, again, is the normal colon. You see there's this A compartment, and there's this I and B compartment. They've got nice, stable methylation, which by the way, as you age, it goes down a bit. As you divide, it goes down a bit. But let's say this is a younger normal colon. Here's the colon tumor. You get hypomethylation. You disorganize. I and B move around. And then these compartments, I and B, they lose the methylation. They gain K27ME3 and K9ME3, repressive histone modifications. And as I told you, most of the genes in these compartments are actually downregulated in the tumors. Okay. So we're going to look at those genes, and I think it's part of hopefully make a case to you that why we think hypomethylation and the associated genome reorganization are tumor suppressive. There's a little trick Martin kind of did by taking all of the TCGA colon tumors. And you can kind of take all the tumors and you can score them by how hypomethylated they are. Some tumors are a lot more hypomethylated than other tumors. And then you can ask, what happens to gene expression? Are there genes that go up when you're hypomethylated? Are there genes that get silenced more in hypomethylated tumors? And this is sort of what this volcano plot is showing here. And you can see, well, as I told you, most of the genes seem to go down with hypomethylation. By the way, we're only looking at genes in the hypomethylated blocks. We're only looking at genes in those regions. Some of them go up. Let me tell you what those genes are. The ones that go up, maybe many of you won't be surprised to see, they're transposable elements, ERVs and lines, and they're cancer germline antigens. So these are the ones when you become hypomethylated, we know they come on, they become de-repressed. There's work from De Cavallo and Cavallo and others that have described Ts kind of coming on in hypomethylated tumors, and of course a role in tumor immunity for the Ts. So those could be immunosuppressive. It could be tumor suppressive. Could be stimulating the immune system. What about these genes coming down? What about the repressed genes? Well, an analysis of these genes in sort of just a pure gene ontology, unbiased look shows that they're involved in stemness, invasion, and EMT. So these are all programs that a tumor may need to engage, right? They may need to engage stemness properties to divide and maintain their primitive state. They may need to undergo EMT if they're going to invade, right? And so the hypomethylation and reorganization is silencing these programs. It's we think reducing the possibility that a cell could undergo EMT or reducing the stemness potential of cells, thus restricting them as they restricting them as they age. Cells divide and they become hypomethylated. They become restrictive, blocking their potential for tumor progression. So if we wanted to sort of get a little more evidence. Of course, it's hard to look in human and ask whether hypomethylation is really protective, but we were lucky that the Grady group at the Fred Hutch had done this very nice study where they'd sampled normal colon, actually. These are biopsies from normal colon, and they took hundreds of individuals. And the first thing I can tell you is that as you look at the individuals across age, you can sure enough see, as we predicted, that as the colon in older individuals age, you can see that it becomes more and more hypomethylated. But even at a given age, there's sort of a range, as you know. So the question is, might the lower methylation, more hypomethylation, is that protective? The cohort of Grady had two groups. It had a low-risk colon cancer group and a high-risk colon cancer group. The high-risk group had concurrent tumors. They weren't sampling from the tumors. They were sampling from another place in the colon. But the high-risk group had concurrent tumors. And hopefully you can appreciate in the data here, we did an analysis of their data. We looked at the methylation in the sort of blocks or in the compartments, those heterochromatic, CBG sparse regions. You can see that the low-risk individuals had significantly lower, had more hypomethylation, and that the high risk that were getting concurrent tumors had less loss of methylation. So this is, not proof, but it's certainly consistent with the hypothesis that hypomethylation, shown here, is protective from tumors. I just want to make sure that the point on aging comes across clearly, right? Hypomethylation occurs with mitosis, I showed you. The more times the cells divide, it occurs in tumors that have divided many times. It's also clearly evident in aging. And many tissues, if you look at aging individuals, there's progressive hypomethylation. So here's the same plot I'm showing you. Hypomethylation, this is again from Ben Berman's nice paper, a gradual loss of methylation over the years in dermis. And I can show you many, many tissues where this is happening. At the same time this is happening, it does look like transposable elements are coming on with age. So you get more TE expression with age. As I showed you, that's what we see in conjunction with hypomethylation. It may be a causal relationship between this DNA hypomethylation and this induction of the transposable elements. As many of you know, and this is, I should be citing a lot more papers here, but induction of TEs can lead to activation of cGAS sting pathway, it can lead to innate immunity, it can lead to interferon-induced apoptosis, it could even lead to adaptive immunity, as I'll touch upon. So this induction of TEs in itself, you can really think of, maybe it's pruning aging cells, but it can also be tumor suppressive and blocking those cells, right? Which leads us to this model here that is going to sort of wrap up the first half of my talk here. Here's a picture of our genome organization, and it's pretty nice and neat with the heterochromatin on the outside, and all the compartments are appropriately methylated at the DNA level. But as cells accumulate divisions, they gradually lose methylation in the inactive compartments. They become hypomethylated. There's this reorganization of the genome topology. TEs get de-repressed. They come on. They drive some degree of immunity. The cells lose stemness. They lose EMT. Lose EMT potential. So in a sense, I have a little T-bar here, and I'm saying this process then, with aging, as cells divide and divide and divide, this is going to be inhibitory and block tumor progression. That's what I'm showing here. A paper just came out that I thought was very, very nice paper and appropriate from Gorbunova lab, where they study blind mole rats, which are different than naked mole rats. I have to get all the mole rats straight. But this blind mole rats, one striking feature, I suppose, is they don't get cancer very often. So the argument is, why don't blind mole rats get cancer? What this paper actually showed was that they have a little bit of a defect in DNMT1, the maintenance DNA methyltransferase. So this paper showed that blind mole rats hypomethylate their genome pretty fast as cells divide. So whatever's happening here in humans and mammals, in blind mole rats, is happening faster. And when that happens, the TEs come on. That paper really focused on the transposable elements and how the transposable elements could trigger an innate immune response and interfere and induce cell death. So this may be a regulatable process across different kinds of mammals that you can, or even across different cell types within an individual, where maybe you can regulate the degree to which you hypomethylate at the time to sort of, walk the tightrope between, needing to self-renew tissues and needing to avoid risk of cancer. Oh, and by the way, this epigenetic mechanism, this counter of mitotic cell divisions, identifying at-risk cells that have maybe acquired mutations because they divided many times. It's very analogous, of course, to a genetic mechanism that you're probably all familiar with, and that is telomere attrition, right? You know, the telomeres shorten as cells divide, and this is the Hayflick limit, and stop cells from dividing. So in a sense, we're arguing here that there's an epigenetic analog here, the mitotic clock, and that mitotic clock is in parallel also sort of restricting cell fates and potential and acting in a tumor suppressive manner. Okay, but you still get cancer, right? We still have tumors. So how do the tumors overcome the epigenetic clock? This slide, I would say, is a little bit of speculation. So I'll caveat that, but we know that certain types of lineages probably lack an epigenetic clock. I think some of you may be interested to see that in Berman's paper, when he looked at myeloid cells, it was like lock on. There was no hypomethylation. Lymphoid cells, yeah, with age you get more and more hypomethylated. Myeloid cells don't. So maybe a myeloid tumors of some type. Maybe they don't have hypomethylation. They don't trigger this tumor suppressive process. It's not an issue. Maybe some pediatric tumors or stem-like tumors also can avoid the epigenetic clock. So that's one possible way you could avoid. You could evade the epigenetic clock. Of course, my lab studied a lot, and I think the last talk I talked about, there's some tumors that globally increase methylation through mutations. There's these IDH mutations, and there's SDH loss, and BRAF signaling is thought to globally increase methylation. So maybe this is offsetting the clock, methylating those regions a little more and preventing, again, this tumor suppressive process. And there may be other compensatory epigenetic mechanisms. And I'm going to chromatin changes. And I'm actually going to talk one about, my next part of my talk is going to be one of those. And I want to talk about amplification of a histone methyltransferase that represses TEs, represses transposable elements. So this might be very important in tumors that, to offset this epigenetic clock for progression. All right. Switching gears a little bit, that tumors go to great lengths to evade the immune system, right? They have inhibitory molecules that, trigger T cell exhaustion. They like to shut off antigen presentation pathways. They, have interferon and cytokines that interfere with immune responses. They like to block these pathways as well, and I want to show this slide because epigenetic regulators are implicated in a lot of these processes, right? PRC2 or the polycomb complexes silence MHC, so they prevent antigen presentation. This is a good way to hide from the immune system, right? PBAF and ASF1 suppress interferon signaling, right? And as I said, the NMTs and actually LSD1 can suppress the innate immune sense, the innate double-stranded RNA sensing pathways, okay? And there's a lot of literature on this. And the project I want to tell you about today is really focused on checkpoint therapy and epigenetic regulators. And we sought to identify epigenetic regulators that might help checkpoint therapy work. To do this, we use syngeneic mouse tumor models. So these are sort of B16 melanomas or LLC lung cancers that can be transplanted into new mice. You can do this with into immunocompromised mice shown in gray, and the tumors grow super fast, and the mice have to be euthanized. Put this into wild type tumors that are immunocompetent, the tumors grow this fast. But if you include checkpoint therapy, of course, the PD-1 like therapies, the tumors don't grow as well because you exaggerated the immune or accentuated the immune response against these tumors that are coming in. I guess the question is, can you take can you do even better in some of these conditions by hitting some of the epigenetic regulators? I'll tell you how we went about this. We did a screen. This is work with Gabe Griffin, who was a postdoc in the lab, is now a faculty at Dana-Farber, Rob Manguso and Kathleen Yates at the Broad Institute. It's kind of a complicated system. Well, it's not that complicated, but it's basically it's a CRISPR screen. So the idea is you take the cancer cell lines, and we've introduced Cas9, so they all express Cas9. And we put then a pooled library of epigenetic regulator knockouts in there, guides against different epigenetic regulators. The library contains guides to like almost 1,000 genes and 6 guide RNAs per gene. So it's a pretty big library. It's infected into these tumor cells, and they expand the tumor cells. So now you have a big pool of tumor cells, and each cell has kind of lost a different chromatin regulator. So you then, you can treat them with interferon in vitro, but much more interestingly, you can put them into mice. You can transplant them into mice. You can transplant them into immunocompromised mice, wild-type mice, or mice treated with checkpoint therapy. And in a sense, this is the most interesting arm. We're most interested in sort of epigenetic regulators that help that checkpoint therapy work better. Or if you knock them out, they become quite resistant here, but that don't really have much. We're not interested in things that are affecting immunocompromised mice, because those are likely intrinsic growth issues for the tumor. For those who don't do these screens, you can grow out the tumors, and then you can pull out the DNA. You just sequence, you PCR up the guides, and you sequence out the guides, and then all of a sudden you get this list of presence of guides. And what we're really looking for are guides that are being lost here in the checkpoint, because that knockout made that checkpoint therapy work way better. And we're interested most in guides that are enriched here, but not in the NSG mice. So we're doing a differential. All right, so this is kind of what this picture looks like. These are guides in immune checkpoint versus not SCID, and to the left are things where they're depleted, right? It means that tumor cell that knocked this thing out was unable to grow in the presence of an immune system. So in a sense, they're drug targets. I guess the first thing that was, I don't know if it's disappointing or just reality, was that we got a pretty different set. This is showing the axis here is for the B16 tumor compared to like the B16 is a melanoma compared to the lung tumor. You can see you get mostly different regulators. So it's not you're getting commonality, right? Different regulators sensitized to immunotherapy in different contexts. Except these pink ones. These pink ones were the ones that were more interesting to us. They're shared targets, and they sensitize, knockdown of those regulators sensitizes to immunotherapy in both LLC and B16. So anyway, you might notice some of the things that you're familiar with from the literature, EZH2 and SMARCA, and most of them are either in one model or the other, but here's this really interesting list that was shared. Now the top hit right here, all the way on the left, was SETDB1. This was really interesting to us. And it was interesting to us because, well, it was the top hit. It's a histone methyltransferase, and it's in these two different complexes. It's a repressor. It's in the CAP1 complex, and CAP1 complex has TRIM28. You can see right here, TRIM28 scoring in B16. And then there's the HESH complex, which is another silencing complex that relies on SETDB1. And these guys scored in LLC, but SETDB1 scores in both, right? And it's interesting to think that it might be acting here in two different complexes, depending on the context. But anyway, it seems quite important. You knock it out, the tumors are really sensitized. I don't want to spend a lot of time. We did a lot of validation. Of course, this is just a pooled screen. So now we did individual tumors and individual mice. And sure enough, this is great. But if you knock out SETDB1, it doesn't, in wild type mice, there's not much difference. It has no effect on the tumor growth. And the same if you knock out TRIM28, the component of the CAP1 complex, no difference, right? But in those mice treated with immune checkpoint blockade, look at that. You knock out SETDB1, the tumors don't grow at all. You knock out TRIM28, and the tumors don't grow at all. We validated all these other complex members too. So there's a lot of components, and they validated other syngeneic tumor models as well. Okay, so that's mouse. What about human? Is this maybe relevant to human tumors? And it turns out that ZTB1 is sitting in this recurrently amplified region that's amplified in a huge proportion of human tumors. Actually, Lenzon found this some years ago, just as a big region of amplification, includes ZTB1. It's really, really common. It's in a huge proportion of tumors, particularly like liver and lung and breast. They all have these amplifications of this region. Now, the caveat is it's a pretty big region, right? There's a lot of genes in the region, so ZTB1 may not be the only important gene. I mean, almost certainly it's not the only important gene in this amplified region, but clearly ZTB1 is amplified. And when it is amplified, its expression, shown here, is up, okay? So ZTB1 is highly expressed in human tumors, and it's amplified in human tumors. And you then might ask, well, does it suppress immune response? And the answer is, well, it actually, at least in one trial, it prevents response to checkpoint therapy. This was a trial that treated renal cell carcinoma with either checkpoint or with mTOR. This is a non-immune pathway. In the case of the mTOR, the non-immune pathway, it made no difference whether you had ZTB1 amplification or not. But in the case of the checkpoint therapy, if you had a ZTB1 amplification, you did much worse. Red. You didn't respond as well to the therapy. Just like what we see in mice, right? In mice, we see if you knock out ZTB1, you respond better to therapy. Here we're seeing that in humans, if you have an amplification of ZTB1, high expression of ZTB1, you respond less well to therapy, which is exactly what we predicted. So what is ZTB1 doing? So ZTB1, it targets big regions. People who think about chromatin and heterochromatin and K9ME3, they tend to be very spread out. It targets regions of tens to 100 kilobases in size. We mapped K9ME3 in B16 cells, either in control or after we knocked out, shown here, knocked out ZTB1. You can see you've lost K9ME3. You can zoom in on a region. Here's one of our sort of a representative region. Nice peak of K9ME3 repressive. You knock out ZTB1, you lose this peak. And look, all these accessible sites come about, right? These accessible sites come about, and CTCF, we think CTCF's binding, transcription factors are binding, and lots of genes come on in their vicinity. And in this locus, you can see there's lots of these TEs, the LTRs throughout this locus. We think ZTB1, we know through literature that ZTB1 targets, Peter Jones and others, ZTB1 targets transposable elements. And you can see that they're being de-repressed in some of these regions. It's also interesting, these regions are also contain a lot of segmental duplication. So this is how the genome evolves. It copies things and it does different things, it copies genes. And we see a lot of these evolving gene clusters as well being enriched among ZTB1 targets. So, all right. So what, genome-wide, among ZTB1 targets, you see LTRs, TEs, segmental duplications. You also see a lot of immune genes are nearby, right? And this makes sense because immune loci are all often very duplicative in clusters, and they're evolving. Your immune system is under high pressure from viral other insults that evolve over time, and so your immune system is also evolving. And we think ZTB1 is playing an important role in sort of buffering this evolution of immune programs. Again, I just want to make the point, this is genome-wide. ZTB1's, hitting a lot of transposable elements. It's hitting clusters of duplicated immune genes, and I could just show you there's many, many, many examples of that. NKG2 receptor genes, they're all sort of de-repressed when you knock out ZTB1. So ZTB1 seems to be silencing both transposable elements and a lot of important immune signaling genes. So I want to hone in, though, because part of the topic here today is these transposable elements and silencing of transposable elements. And ZTB1's acting, we think, mainly in compartment A, not in the heterochromatin. So it's a little different than what I talked about in the previous part of the talk. But it's clearly silencing and suppressing transposable elements. And this is just an example of showing that all these TEs come on in LLCs when you knock out, at the RNA level, when you knock out ZTB1. in B16, all these TEs here are coming on, they're being induced. And if you kind of squint a little bit, you can appreciate what's special about all these TEs that are coming on. Well, they are young TEs. They're young TEs that have retained their viral ORF. So not only are they making RNA, and they could be leading to this double-stranded RNA response, but I think in these models, more importantly, they're making peptides. They're making parts of the envelope in these viral relic genes. And in fact, we can see this at the peptide level. We see that there are these TE-encoded peptides, these sort of viral ORFs. Not only are they being expressed, but they're being expressed and they're being presented by MHC on the surface of the cancer cells, being presented to the immune system. So when you knock out CETB1, you are turning on all these TEs. These TEs are being presented to the immune system. And better than that, we can actually isolate T cells that we know recognize these MHC-presented TEs. And we know that when you knock out CETB1, the numbers of those T cells that recognize TEs goes way up. And the sort of cytotoxic index, those T cells get much more cytotoxic and activated when you knock out CETB1 because they're responding to this increased expression and presentation of these TE encoded peptides. Let's see, I'm not showing it here, but I can just tell you, we've shown that for the CETB1 knockout to stimulate the immune system, to stimulate this response, you actually need MHC presentation. If you knock out MHC, you don't get this response, and you need the T cells. So not only am I kind of showing you that the T cells are getting activated and that the peptides are being presented, we have other data to show that actually both those events are essential for that enhancement of immune therapy, of checkpoint therapy in the CETB1 knockout. Okay. All right. So good. I think I have a few minutes for questions. So this part of the talk, I told you, CETB1 is, we think, a really interesting immunotherapy target. It was the top hit in a screen for epigenetic regulators that sort of confer resistance to checkpoint therapy in mouse. We know that it's amplified in a lot of human tumors, and that when it's amplified, it seems to be associated with the resistance to checkpoint therapy. So we think it's a really interesting drug target for that region. Mechanistically, I showed you that CETB1 silences transposable elements, and it also silences a lot of immune loci that are packed with transposable elements and are duplicated and seem to be evolving. And we think there's probably a very interesting role being played there in sort of buffering evolution of the immune system. And finally, I think I gave you some direct evidence that TE-encoded viral ORFs on cancer cells can activate T cell immunity. And I think that kind of pertains back to also what I was talking about aging cells. Aging cells are probably also expressing some TE-encoded ORFs. And so it's an interesting thing about what happens to immune response in aging individuals. But clearly we can see some of this in the tumor models. Okay, so I will make an attempt now to tie together the different parts of the talk. I told you about the mitotic epigenetic clock where cells lose DNA methylation, become progressively hypomethylated, as they accumulate divisions. This is the mitotic epigenetic clock. I kind of speculate that I think it's a deterministic underlying component of the aging clock that we've heard a lot about. Although I think that second piece is a little bit speculative. How much of the aging clock can really be explained by a mitotic hypomethylation clock? Nonetheless, hypomethylation clearly happens as cells divide. Clearly happens more and more in aging tissues, and I hopefully have convinced you that it's probably tumor suppressive. It's leading to the induction of TEs and immunity. It's reorganizing the genome in ways to restrict cell fate and prevent them from being tumor-like. And I speculated a little bit on how tumors might avoid or might overcome this barrier, this epigenetic barrier of hypomethylation. And then I showed you one example of how if you upregulate, amplify a chromatin repressor, you can silence TEs, and that something like this may allow tumors to both clearly evade the immune response, and maybe overcome hypomethylation processes. More broadly, I hope this gave you some ideas, and that the epigenetic clock probably has a lot of clinical opportunities, both in aging and in cancer, that we could all think about. Sarah Johnstone led the genome topology work. Sarah was in my lab. She's now a assistant professor at Dana-Farber. Martin Ari is a terrific biostatistician. He's a professor at Mass General. He's collaborated with us. Gabe Griffin, as I mentioned, is now a faculty at Dana-Farber. Jingyi Wu is a terrific computational biologist in my lab. Rob Venguso is our collaborator at the Broad and MGH and the tumor immunity work. Thank all the members of my lab and our collaborators at the Broad and MIT, Dana-Farber, and all the sources of funding. And thanks for listening, and thanks, Paul.
Thank you, Brad. That was an outstanding talk, and I'm sure we have plenty of questions. We have a few minutes before the hour, so we can jump right in. And then, as mentioned, Brad has kindly offered to stay past the hour for additional discussion. And so I'm going to start it off with, I think, a question related to the first part of your talk. If hypomethylation of tissues increases with age, and theoretically hypomethylation has a protective effect from the point of view of tumorigenesis, how do you explain increasing incidence of cancers in general with increasing age?
Well, there's this balance, right? As cells divide more and more times, they accumulate mutations. The epigenetic clock is trying to offset that. It may be that as you age, the epigenetic clock and the induction of TEs is less effective, because I bet you a lot of older people are walking around with a lot of expression of TEs, and they must at some level be tolerized. The immune system must be tolerized to those TEs. So it might be that as you age more and more, eventually the epigenetic clock is not as effective, because your immune system is not going to, is a little bit tolerized to those TEs. And but those mutations, they're still accumulating, right? As we know from much work, both in the cells are accumulating mutations as we age. You can talk about all sorts of recent genomic studies in addition to everything we knew in the past. So, it's kind of, there's a little match, a battle going on, right, between the mutation accumulation and then the telomeres and the epigenetic clock trying to stop it.
Very good. So I had my own question about gene density within the TADs that are subjected to this hypomethylation. So I think when I think of hypomethylation in cancer, we think about that affecting gene deserts, but you clearly showed examples of genes that are differentially expressed or being repressed and associated with this hypomethylation. So is there a way to generalize it, or can you speak to?
You're right. Let me
to that paradox.
You're totally right. Those hypomethylated blocks are gene poor, very few genes, but they're so massive and they make up, 30-40% of our genome. There's actually a lot of genes in there, even though they're very sparse and few and far between. There's still quite a lot of genes in the hypomethylated block. It's just that the compartment A, the active U-chromatin, those are packed with genes, and there's far more genes in there, but there's still more than 1,000 or so genes in the gene deserts, just because they're so vast. Right. But you're absolutely right. Those are the places where this action's going on.
Okay. Okay. So another question from the audience: would metastasizing tumor cells or cancer stem cells that upregulated EMT and stem cell genes, would they have differential methylation and histone modification landscapes compared to what you showed where hypomethylated regions become more compact?
That's a really interesting question. So we were thinking about this a lot, that these regions are getting hypomethylated and chromatin is really shutting them down, and it's going to prevent EMT. But we know EMT goes on, so they must be, this must be overcome in some ways. I pulled up a paper. There's a paper from maybe 10 years ago from Andy Feinberg's lab that describes the EMT process requiring the remodeling of K9me3 heterochromatin. So I bet the questioner is right. I bet that there's some remodeling going on in conjunction with EMT, and that the process is trying to prevent it from happening, but there's if there's the right mutations to trigger the right signaling pathways, there's certainly a selective advantage if some cell acquires that ability. There's a selective advantage in metastasis and tumor. So that's a good question. I bet there is remodeling.
Some interesting follow-up work there, obviously.
Yeah, I think there is.
Yeah. Okay, so it's the top of the hour. I'd like to thank Dr. Bernstein for his wonderful talk and to officially close the event. But those who are interested to stay on for additional discussion and Q&A may do so. Thank you all for coming, and please join us next week, October 21st, when Dr. Minyoung Lo of UCSF will be presenting on what will it take to cure every child with acute lymphoblastic leukemia. So that will close the official event, and we'll continue on with an extended Q&A. All right, what else do we got here in the chat? So set DB1 sites are predominantly in compartment A of normal cells. Is there a difference between the predominance of those sites in compartments of dividing and non-dividing cells?
Oh, interesting. I mean, that's an interesting question. So the question is, in like tumor cells, does this change versus post-mitotic or not dividing cells? God, that's a really interesting thought. I don't know the answer to that. I mean, I do think that there's an interesting possibility for variegation, and that if you lose the repressive marks there, the heterochromatic state, you're on for a little bit, and that could be heritable through mitosis. And so you can imagine that there's almost like an epiclonality of like de-repressed or repressed. But I can't get my head around exactly what would happen. I suppose that would require proliferating cells. So I don't know what would happen in post-mitotic cells or non-dividing cells.
Very good. So I had another question. Perhaps this is another future direction or something that remains to be investigated. But you showed the really nice example in colon cancer versus normal colon tissue, where there's the preponderance of hypomethylation in the colon cancer. Given the beautiful work that's been done for many decades now by Bert Vogelstein describing this sort of linear progression, the Vogelgram, from a normal colon epithelium to an early adenoma, late adenoma, and then a frank colon carcinoma. Is there any opportunity to look at what are the kinetics or their specific time points during the evolution of these tumors from normal epithelium to pre-neoplastic lesion to a bona fide tumor, and how, just what's the general sort of accumulation of this hypomethylation? Does anyone know, or do you have any?
That's a great question. Actually, it's a great question. We first of all, it's a great question, and you want to look at adenomas. It is really hard to get your hands on adenomas because the pathologist has to take an adenoma and look at every square millimeter of that adenoma to make sure there's no actual malignant tumor there. We got some adenomas, though. We got a couple examples, and sure enough, the adenomas have an intermediate hypomethylation. I wanted to look at their high C maps and their chromatin structure to see if they were intermediate in terms of this disorganization. We couldn't do that. But they're clearly intermediate in terms of hypomethylation. They haven't quite gotten to that really massive hypomethylation at tumor, but they're more than a normal colon cell. So yeah, so it's consistent with the idea of this progression is more and more divisions and this accumulation.
Great. So I think I've covered pretty much there were a lot of redundant questions about the so-called protective effect and associated with the hypomethylation. I think we've covered pretty much all of those. So if there are any
new one, and something about IDH mutant glioma.
Yes, in the context of mutant IDH glioma, inhibiting hypermethylation does not inhibit tumor growth, despite the hypothesis that enhancer insulation is involved in tumorigenesis. What are your thoughts?
I'm curious how we know that for certain. Because if you treat, how you know that inhibiting hypermethylation, I see. Yeah.
With the demethylating agent, or
I didn't think that there's a question here. Maybe it's about a different topic a little bit that we've worked on, but the question is, we've argued in other stories that hypermethylation displaces an insulator and activates PDGFRA. And if you treat a cell line with IDH inhibitor, this is a complicated question. So I would think if you treat a cell line model with demethylating agent, you probably do stop proliferation because you can remove the methylation. It's very hard to do clinically. I could talk about that, why it's so hard to remove methylation with demethylating agent clinically, because it's slow cell divisions. But now the person changed the question and clarified that actually don't mean methylation, they mean IDH inhibitor treatment. I think IDH inhibitor treatment probably should work, but you just have to go very early. Once you have a profound change in methylation, it's going to be too late, right? Because the methylation is mitotically heritable, as I showed you. So if you're going to interfere with something that alters, that is an initial stimulus that alters the methylation, you're going to have to do it very early. So I think that IDH inhibitor might work, but you'd have to go in very early, because if it's later, there's a hit and run. The cat's out of the bag already. You've already hypermethylated the enhancer, the insulator, and it's probably too late. That's my speculation of you just have to go early.
Right. Okay. Well, I think we'll wrap things up then. We definitely appreciate all of your time, Brad.
There's one from Charlie.
Oh, I don't have it yet.
Really?
Here it is. He mentioned the relationship of hypomethylation of gene deserts versus telomere shortening. Thoughts on why both are needed as mitotic clocks. Does demethylation in gene deserts trigger apoptosis? Or trigger apoptosis genes, rather?
Good question about why you need both. Why do you need an epigenetic clock and an epigenetic mitotic clock and a genetic mitotic clock? I wonder if the epigenetic mitotic clock, here's why I think you need them both. I would speculate the epigenetic clock can be, remember I told you that you can up or down DNMT1, and that, in the blind mole rats, DNMT1 is a little hypomorph, so you age quicker. I bet you that methylation, the hypomethylation, is more regulatable across tissues and might even be able to reset in a stem cell. So I would think that's part of the answer. That'd be my speculation, but it's probably not. Why do you need more than one tumor suppressor? There's lots of tumor suppressor genes. Why don't you just need BRCA1? I don't know. Maybe they're a little redundant mechanism. But it's a good question. I'll think about that more. What's different about it? I bet you that the epigenetic one is more regulatable.
Okay, it looks like there's one more question that's also popped up here. Do tumor cells remove CpG methylation and prefer histone methylation because they are potentially more controllable or dynamic?
I guess my hypothesis, or our hypothesis, is that they remove that the CpG methylation is being removed because of the dividing, and the chromatin, the histone methylation, is coming in there as like a backup to keep these genes off. It's probably, I don't know that the tumors are trying to do it. I don't think of it as the tumors trying to do something. I think of it as really just a hierarchy of epigenetics. It's like when you lose the methylation, then the CpGs are available, and they're not adherent to the lamin, then the repressive chromatin can get at it. And there's physiologic consequences to that. Like I showed you, the silencing of EMT genes and things like that, but also probably, I don't know if I want to say, but if you go look at the senescence literature, you'll see a lot in the senescence literature about how those histone methylation changes could be contributing to senescence as well. So yeah, I guess that's my answer.
All right. Okay, let's wrap up then. So thank you again, Brad, and definitely appreciate your time. And it was an outstanding turnout today. So we'll see you again soon. Thank you, everyone, for attending.
We'll see you in Memphis sometime soon, hopefully.
Sometime soon. We better. See ya.